All right. Hi, Kara. Thanks for joining us. My name is Lisa, and I'm at the Saskatchewan Science Center. I'm so glad you could join us today. I am honored to be here with Kara Loos, Dr. Kara Loos, I should say, who has been working at the Roy Ramino, did I say that right, provincial lab during COVID, and is a microbiologist working at the Institute for Microbial Systems and Society. Welcome, Kara. That's a mouthful to say, hey?
It is, yes. Thank you for
Awesome.
having me.
Well, thanks for joining us today. Maybe we can start with just talking a little bit about your work at that provincial lab.
Sure. So my primary employment is at the University of Regina with the Institute for Microbial Systems and Society. So I'm both a lab manager as well as a research associate, which basically just means I get to do a lot of really cool research.
That's
When the
awesome.
pandemic started, I was recruited back to the Roy Romano Provincial Lab to help with COVID testing and research. So the work that I've been doing over there since the pandemic started initially was just kind of getting testing up and going, trying to deal with the overwhelming shift in activities there. So before the pandemic started, we weren't even doing COVID testing, and all of a sudden there was this huge need to have to test for hundreds to thousands of people immediately. So just helping out that team to get that going and validating everything was really where it started. And then over the course of the pandemic, that evolved, obviously, the variants being a very hot topic in the media recently. So just trying to get those efforts up and just really being able to pivot and shift into what the demand is. So my role over there has really been all COVID focused, but really just trying to get that going. And then really just there's still the respiratory season. So trying to help out with testing in those other areas as well. Did you want to hear about the U of R stuff right now as well, or just
Sure.
okay?
Yeah.
So I work in a variety of different projects. The Institute for Microbial Systems and Society, or the acronym IMSS. So I'll just use that from now on, a
Perfect.
little bit easier. We deal with a lot of different projects. So anywhere from looking at the microbial communities in bees and how that could be affecting their decline, all the way up to COVID testing. We do a lot of just a huge variety of different collaborations. So anywhere from environmental to obviously clinical with the case of COVID-19. Our focus is primarily genomics, so looking at the genetic properties and specifically the microbe interaction. So whether that be like a fungus or a bacteria or a virus, we like to look at the genetic properties and how those can be affecting those interactions. So really, since the pandemic started, my biggest project has been working with provincial health labs. So for this project, we are working with the provincial lab in Saskatchewan, so the Roy Romano Provincial Lab. We're also working with the BC CDC in BC and the Cadham Provincial Lab in Manitoba to get patient samples that were originally positive for COVID-19 sent to us. And then we were looking at sequencing the COVID-19 genome and as well looking for co-infections. So maybe a patient that's infected with COVID-19, are they also infected with other kinds of respiratory viruses, which has been documented in the
Hmm.
past, that maybe somebody might have COVID as well as influenza or RSV, which can be serious in themselves. But what happens when you're infected with both viruses? Does that result in maybe a more serious effect? So when the pandemic started, provincial labs were very quickly overwhelmed with testing. And maybe, they weren't able to then test for COVID-19 and a huge array of other respiratory viruses, which in the past they would have done. So everything was COVID, 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 but maybe some of those COVID patients that have this very serious result, maybe they also had some other kind of respiratory virus. So that was kind of the nature of our research, was to investigate this area to see maybe how these interactions were happening and how that might be affecting patient outcomes. So not only were we looking at that, but we were also just developing genomic protocols that researchers around the world have been doing, but just trying to find these protocols that you can quickly and effectively pull out these genomes to look at, let's say, variants or changes that could be resulting in the effectiveness of vaccines, maybe if a mutation occurred in an area where we're testing, that could result in a false negative, and even just getting that information for treatments and vaccine development. So that kind of information could come out of this research, and just as well as the other respiratory viruses circulating at that time. And then also on top of that, we have a collaboration with a program called PREDICT, which is a global program that works to survey pathogens that can then spill over from animal hosts to people, which a lot of us know a lot about that now with COVID-19.
Yeah.
So we were working with PREDICT before the pandemic started. So we like to joke that we were working with coronaviruses before they were cool. But we got these bat swabs just before the pandemic started so they are bat swabs from African bats. And they found that these bats were carrying novel coronaviruses. And so coronaviruses we didn't know a lot about, that their sequences were not yet available globally. So they sent us these swabs, and our job was to try to sequence these new coronaviruses. So we've been working on that as well, in collaboration with PREDICT, to just get more information about coronaviruses.
Wow, fascinating. Absolutely fascinating. That's incredible. Good for you. Okay. Do you mind if we start at the basics and just talk about what a virus actually is, and the difference between, like you say, novel coronaviruses and viruses we've had in the past that are not coronaviruses?
Sure. Yeah. So we'll start with then, your question of what is a virus. So they're very small. You can't see them with your eye. You need a microscope to be able to see them. And when you do look at them, they're pretty simple compared to a lot of other organisms. So viruses are infectious. So we know with coronavirus specifically that can be infectious to us, but viruses don't just infect humans. They can infect a variety of different hosts. So like bacteria, plants, or other animals. And a big topic of debate between scientists is whether viruses are actually alive, because a component of viruses is that they can't survive for very long without a host. So in the case of COVID-19, we're the host.
Yeah.
So they can survive on surfaces, and there's lots of education that has gone out to, disinfecting and how to prevent transmission. But ultimately, they can't survive on these surfaces for very long. So they do need these hosts, and that is part of their process. And then the genomes of viruses can vary significantly. So they can have DNA or RNA. A lot of people have heard about RNA in the news with the vaccines. They can be single or double stranded. They can be linear or circular, and they have huge varieties in the length. So viruses are very diverse, and a lot of that has to do with their host and how they transmit. What else would you like to talk about? What's your next question on that?
I guess viruses have, like the coronavirus itself has evolved to have like the crown and the protein. And I guess what is my question? My question is, why is the coronavirus so contagious? Is it because of its shape or its genomic properties, or?
Yes, let me get to that.
I jumped around. I apologize.
Okay, so specifically then with COVID-19, it is a coronavirus. A lot of people have heard that. A lot of people have never heard of coronaviruses before, when actually coronavirus is one of the common colds. So a lot of people would say in the past, oh, I got the flu, when actually there's several respiratory viruses that are circulating in what a lot of people call flu season, when it's actually a respiratory season. We have a lot of different respiratory viruses circulating. So there's 4 to 5 main coronaviruses that a lot of people were probably infected with it without even knowing with it. It was often just called the common cold. These coronaviruses are then a family of viruses that can cause illness. So I already mentioned the common cold. It can also cause the severe acute respiratory syndrome, or SARS, which is the acronym we use with COVID-19, or SARS CoV-2. And coronaviruses can also cause the Middle East respiratory syndrome, or MERS. So SARS CoV-2 specifically shares both similarities and differences with these other coronaviruses. It's more contagious than other coronaviruses like the common cold, initially likely because it was so new to humans. So we didn't have anything like a herd immunity. or any kind of information to be able to combat the virus initially. It also seems to spread more easily than other respiratory viruses, which can then, um, obviously, it can spread a lot faster. It also seems to be a bit more contagious for a longer period of time. So initially, a lot of the things were that you could have COVID-19 before you're even, and be contagious before you even have symptoms. So right
Hmm.
now, we're saying, monitor, self-monitor, don't come into work if you're exhibiting symptoms. So you might end up going to work and spreading the virus before you even know that you have that virus. So that can also cause to it being more contagious. So yeah, initially we didn't have a way to prepare for it. Our immune systems were not ready to fight it.
Hmm.
This can result then in the virus causing more cellular damage and additional complications. Those complications are still being researched and a lot of information is coming out of that. But for, especially for people with decreased immunity due to either medications or specific conditions, or even just from aging, the resulting COVID-19 disease can be more serious for those individuals.
Hmm. Interesting. And picking back on what you said in your introduction to those complications, like having different respiratory diseases with COVID, that's such an interesting thing to research. And yeah,
Well, if you think about it, so yeah, just talking about, immunocompromised individuals. So when you do
right.
become infected, let's say with COVID-19, that then makes you immunocompromised because your body's working very hard to fight off this infection. Well, what
Hmm.
happens now when you become also infected with influenza and also RSV? It's like,
Right.
just imagine your immune system trying to combat all of those viruses at the same time. I know,
Yeah.
working at a provincial lab, I've seen individuals who have, we used to test for a panel of different respiratory viruses, and I've seen people infected with 5 different viruses at one time. So, you know,
Wow.
those are all in different stages, like maybe you're just getting over one coronavirus, but you were then infected with RSV. And so maybe it's not super overwhelming, but what if they're all, a very active infection at the same time? That could be a more complicated case then for that individual. So yeah, it's very complicated, and being infected with one virus doesn't make you immune to all the other viruses. In fact, it makes you, in theory, more susceptible to those infections.
Right. Interesting. Wow. Okay. You had mentioned RNA before, so maybe we could just talk a little bit about the difference between DNA and RNA, because you're right, we have heard a lot about RNA in the news lately because of the vaccine. So,
Yes. So I mean, I can talk about the broad differences between DNA and RNA. Like, DNA contains sugar deoxyribose, while RNA contains the sugar ribose. So that's, where
okay.
the DNA stands for deoxy, like the D stands for deoxyribose, and the R stands for ribose. I mean, those are your general terms. Traditionally, DNA is more of a double-stranded molecule, so you see that double helix. In our DNA, versus
Mhm.
RNA is usually a single-stranded molecule. But specifically, what's coming up in the media is RNA with respect to viruses. So in our bodies, we have DNA and RNA all throughout our cells, and what DNA and RNA do in our cells are have very different responsibilities.
Mhm.
Ultimately, RNA is not as stable as DNA. Meaning, so if it's not as stable, that means it can change easier. And this
Right.
becomes more relevant in the case of variants. So in the case of COVID-19, it is an RNA virus, but there are DNA and RNA viruses. There, you see both. Um, so yes, DNA and RNA have different functions in human. DNA can be responsible for storing and transferring genetic information, versus RNA is directly coding for like amino acids and act for like a messenger to make proteins. But in the case of
Mhm.
viruses, which can be either DNA or RNA, DNA viruses have to change this viral DNA into RNA to make proteins, versus RNA viruses can skip this process. So it's a lot faster. RNA viruses have these higher mutation rates because of this, and this provides a selection for faster replication, so it can make copies a lot faster in your body. But then these can also result in mistakes. So viruses will integrate our cells, and manipulate our natural mechanisms to make copies. And then they kind of burst out of the cell and lyse our cells. Lysing just means bursting the cells. And then they can reinfect another host or just replicate within your own body. So during that process, there's mistakes that happen within the genetic code. And those mistakes aren't necessarily done on purpose. So there's just maybe it's being done very fast. It's like if you're trying to write something out really fast and you make a spelling mistake. That's like what happens during the process. Now, these mistakes or changes can be beneficial or harmful. So harmful could be that it makes a change and now it just that strain becomes extinct because it's no longer able to replicate, for example.
Right.
Or it can be beneficial where that mistake or that change now makes that virus more contagious. It's now changed, let's say, the spike protein, which, you know, a lot of people have heard a lot about. So now it's more effective at causing disease. So these mistakes happen, not necessarily on purpose, but it is how viruses evolve, and it is a natural process. Variant strains have become very popular in the media. This is how viruses work. This is how they evolve. It's not new to viruses. It's just we're all intimately invested now in how these variants are interacting and affecting the global response to the pandemic.
Interesting. So those mistakes that you're talking about, I love that analogy. I think that's great. Like how often would you say these mistakes happen? Like variants are produced because of basically they're mutating, I guess. Is that correct?
Yeah. How often?
Yeah.
I don't have an exact rate, but it's constantly. It's constant. Yeah, when in the data set that I was looking at for COVID genomes for the prairies, you know, we're seeing I think I only saw one genome that didn't have a single change from the original Wuhan strain. Like they're constantly changing. So there's in all of them, you know, one might have as many as 20 different changes or maybe just a couple. But a lot of those don't really matter because it's not making it more contagious. They're just changing. They mutate very quickly. And we've heard about this in the past with the influenza virus, why there has to be a new vaccine every year. When in actuality, there's different types of influenza. So there's influenza A and influenza B. So A has like your H3N2 or your H1N1, which a lot of people know about because of the more recent pandemic with that strain. But the influenza A virus mutates 3 times faster than the influenza B virus. So if you get an influenza vaccine, it has both strains for, you know, the H3N2 influenza A, your H1N1, also influenza B. And even if it's not a match for the, you know, circulating H3N2, which mutates very quickly, the influenza B does not mutate very quickly. So even though you might not be protected against the one strain, you will be likely for the influenza B. So it's very complicated on how quickly they mutate, and different strains mutate at different rates. And I don't think we really know that yet for COVID-19. But how often viruses mutate, it's constantly, especially with single-stranded RNA viruses. They're so simple and quick to replicate that these changes, there's not as many proofreading mechanisms in place to fix those mistakes. So they happen very quickly.
Wow, fascinating. Let's talk a little bit about how people get tested for COVID-19 or actual SARS CoV-2, right? Is the actual virus name. So COVID is the disease, right? So how do we get tested? How does that work?
Right. So I mean, I can start from square one. You get your test. Traditionally, there's different swabs you can get. So some people have done just a throat swab or a nasal swab, but the most effective swab is a nasopharyngeal swab, which if you've had to have it done, some people call it a brain tickle, which sounds awful, but really it's a swab that goes up through your nose and touch the back of your throat. So it touches your pharynx at the back of your throat. So it goes in, it's pretty invasive, but the idea is that's where the SARS CoV-2 or COVID-19 those viral particles are going to be present there. So with that swab, it's going to collect out the viral particles. That then gets put into a liquid called a transport medium, which basically just protects the virus particles while it's being shipped to a detection facility. So at the start of the pandemic, primarily all tests in Saskatchewan were being sent here in Regina at the Roy Romano Provincial Lab. Then what they do is they kind of mash up that sample, they get out the viral particles there, and they do a test called PCR, or more specifically qPCR. And it's a special type of PCR, also called quantitative PCR, that can actually, it's very specific to detecting the amount of viral particles in that sample, or also called viral loads. So how it works is that you'll have parts of the virus in your body, and it uses synthesized DNA targets called primers and probes, that it will take an area of the virus that in theory should not mutate very quickly. So it's called a conserved area, that we want to target an area where hopefully there's not going to be a lot of changes. And the reason for that is that these primers, then if the virus is present in that sample, the primers will see a similar sequence and they will bind onto that. So they'll say, hey, yep, that's the sequence that that's the sequence that I can copy. So it binds onto the viral target, and then it makes copies of itself. So the more viruses that are there, the more copies that are going to be made. And every time a copy is made, it releases a fluorescent component. So like a light goes off every time a copy is made of that virus. We can then detect that light. And so as it's making more and more and more copies, you'll see kind of a graph go up where it will just kind of reach like an exponential phase where lots and billions of copies are being made of that virus. And then eventually the test kind of just finishes, and that's that. So if the virus is not present in that sample, those primers do not bind to anything, meaning no fluorescence gets released. So you end up on the graph just seeing nothing because there's no fluorescence being produced. So it's very specific. Now you can imagine that if there's mutations in those regions, those primers may not be able to bind, meaning it can result in a false negative result. And this is something that keeps us all up at night, and we're constantly monitoring, and why it's so important to monitor the genetic sequence and look for changes to make sure that they're not happening in those regions. And just based on the research I've been doing, luckily, we haven't seen changes in those areas yet. So, but it keeps us on our toes to make sure that we are using the most accurate methods in place so that we aren't missing those positives. But that's essentially how the test works.
That's fascinating. That's really cool. I had no idea about the fluorescent pieces. That's cool.
Yeah, traditional PCR doesn't use a fluorescent component. It just basically makes a lot of copies, but this qPCR is very specific. And if you design the test a certain way, you can even say, hey, you have this many copies of that virus in your body. That's not being used that way. It's more being used that, you know, you might see that peak coming up earlier. If there's more of the virus in your body, those copies are happening faster, versus if you're late in your infection and maybe there's not as much virus in your body, that will start happening later. So we can see the difference between like a strong positive and a weak positive. But right now we're not using it in a way that can say, hey, you have this X amount of virus in your body, but you can use it that way too.
So when we actually start showing symptoms, if you are infected, do you have to reach sort of a saturation point, if you will, of viruses being reproduced before you start showing symptoms? Is that why we can pass it on when we're asymptomatic? Sorry. Does that make sense?
Right. That's a really good question. So there are limitations to the test. So if you've had to go through this process with a pandemic, they'll often say, okay, wait a couple days. Let's say that you're a close contact to somebody. Oftentimes, you know, they'll just tell you to self-monitor. But if you're, you know, at a high risk, they might say wait 2 to 3 days before you go to get tested, quarantine, and then go get tested. So the idea is that maybe initially upon infection, you only have very low numbers of that virus in your body. So it's very limited in that it will start to make copies. It's very sensitive, but it might not make enough to kind of get to that area where we can see enough fluorescence. So you want to wait until you have enough virus in your body so that it's able to make enough copies for us to be able to detect it. So it doesn't mean you don't have the virus there. It's just a limitation of the technology. Similarly, towards the end of your infection, the viral particles in your body, maybe you're feeling 100%, you don't have any symptoms anymore. That virus might not really be there in large quantities. In fact, there might not even be a whole viral genome. There might just be bits and pieces that are left over. In that case,
Mhm.
the bits and pieces that are left over, what if those bits and pieces are not part of the area we're targeting? So that can also just show a negative result if there's not a lot of virus left in your body. So there are limitations to it for sure.
Interesting. And then
Does that
as
answer your question?
yeah, I think so. Yeah, yeah. Um, I think as our immune system fights pathogens like viruses and that kind of thing, um, obviously we produce antibodies. So that has also been a kind of testing, hasn't it, in terms of detecting COVID?
So I know one of your questions was, why has rapid testing been slow to catch on in Canada?
Right.
So there's a lot of opinions about this. Um, some people think that the opinions that the health officers are being overly cautious um, on whether promoting too much testing will encourage risky behavior. So, is getting a false negative result more dangerous than no result, kind of question. So,
Mm,
rapid tests do have some limitations in terms of sensitivity. So, all tests have limitations. And I talked about the ones with the qPCR.
right.
In the case of rapid tests, they do not go through this amplification step where they're making copies upon copies. They're just doing like a quick, is that antigen there, like the viral particle? And this comes with a trade-off for sensitivity. So it might not be as selective in being able to test for the virus. So the rate of false negatives is higher. So
Ah,
again, comes down to that, if somebody gets this negative result, hey, I'm going to go visit my immunocompromised grandma. Is that
right.
more dangerous than, just assuming you could be infected? So I think that's where the hesitancy has come. And this whole pandemic has been a really interesting human experiment, really, because all
Mhm.
different countries have responded differently. And it doesn't make it good or bad. It's just people are trying to do the best they can with the information they have. And I think
Absolutely.
that's one of the cases why we're at a different stage with the rapid tests in Canada. But ultimately, these rapid tests are being deployed. Um, so yeah, these rapid tests will never really match the traditional testing methods that are currently in use, like at the
Mm,
provincial lab, which are more sensitive. But the advantage of them is that they are generally cheaper. They can be done outside the lab, so in more remote areas, such as up north, which a lot of
mhm.
times those samples take days to even a week to get, down to Regina, which how helpful is that for people's results? It's not very great when you're waiting over a week for that negative result. Now you've had to quarantine, you've lost income, that kind of stuff. So,
Yeah,
um, so they're really good for screening to people in remote areas that you're worried about. So there is the benefits to them, but again, there is that trade-off that you do have a reduced amount of sensitivity. The
mhm.
other thing too is just availability. A huge battle with the pandemic is that the entire world is looking for the same resources, the same
Yeah,
products, and it's this constant battle. Oh, we're running out of gloves. Oh, we're running out of pipette tips. Oh, we're running out of this, and it's just, where do we secure our next thing? And in the past, companies are like, oh yes, like we'll take your money. But right now, it's everybody wants everything, and they only have so much supply. So it's trying to balance. Do we actually need these versus should we be investing money in the more sensitive test when we're actually able to still get a result around in a day? There's a lot of factors at play, and it's very complicated. I feel bad for those who have to worry about that kind of stuff. But yeah, it's complicated for sure, is the
yeah.
take home message on that. So we talked about then how the PCR test works. So you're getting like a respiratory sample with that, nose swab. Versus antibody testing looks specifically at blood samples. So you would take blood from a patient, and then you're looking specifically for those antibodies that are compatible to the disease in question. So these
Mhm.
antibodies are proteins that are produced by your immune system in response to an infection. So the idea is that if you're infected with COVID-19, you will produce these antibodies, and then they can be detected. To be able to detect these, it usually takes around 2 to 3 weeks for those antibodies to be at a level that we can detect within your body. They can then be detected in your blood for several months or even more after you recover. And that's still unknown because we don't know a lot with, we don't know a lot about COVID-19. We know it can maybe be present for, 9 months or however long. People have been looking at this, but we still don't know exactly how long, but we do know it can persist in your body. There's also research to show that the stronger the initial infection can result in a stronger immune response. So let's say you get a lot of viral particles into your body or a higher viral load, maybe you become a lot sicker than somebody who's asymptomatic, meaning that your immune system produces more antibodies and has that stronger memory for if you become reinfected with it, versus somebody who's maybe asymptomatic, maybe they only have a few viral particles and you don't have as strong as of a immune response for antibody production. So maybe you might become more, or maybe you might be more susceptible to become infected again. I'm saying a lot of maybes here because we still don't know a lot about that, but there's, a lot of things at play, how those antibodies are produced in your body. But essentially, there are tests that can detect it. For the test, these blood samples are then incubated with viral antigens, so with the virus. So they're put with the virus on an assay platform, and there's various different technologies for this. But essentially, if the antibody against the COVID-19 virus is present in the blood, it will bind to that viral antigen, and then you'll get a positive result. I get a lot of questions, or I've heard a lot of questions on why weren't we doing this earlier? And so a lot of questions come to what's the usefulness of getting a result telling you that you've had COVID-19? So let me digress for a second.
Okay.
There's
Yeah.
two
Yeah.
different two different tests out there. So one can tell you, do you have the virus right now? And that's the PCR test.
Right.
This antibody test is telling you, did you have it before? And the usefulness of this comes into play with immunity. So we've seen with measles, which has been called the most infectious virus on the planet. When we had an outbreak, was it 1 or 2 years ago? I'm losing track of time here. When we had a measles outbreak, there was a lot of cases. I remember we had a case at the airport, and so a lot of people got anxious. So all close contacts to that initial case were then screened for antibody testing to see if they had immunity prior. If they did not, then they got a positive test, because we know a lot about measles already. We know how immunity works and how stable it is. For COVID-19, we don't know. You could have those antibodies, but still get infected again. So giving somebody that result, 6 months ago that, hey, you had COVID-19, again, could be dangerous because they might be like, ooh, I'm immune, and go out and infect a whole bunch of people when they become reinfected. We
Right.
also didn't know that even if you're immune, how contagious are you? So let's say that you've had COVID-19 before, you come into contact with it again, but your body has antibodies, so it's fighting it off. But while it's fighting it off, can you still transmit that virus to other people? We didn't really know. That information's starting to come out now that, your contagious rate is decreased after being infected or after being vaccinated, which is great news.
Yeah.
But 6 months ago, we didn't know that. So
Right.
this is a huge reason why we were not doing antibody testing or serology testing at that time. But I think as we gain more information, that's going to become more relevant down the road.
And as more and more people get vaccinated in the province and across the world, that's really important that people understand that if you've had COVID, you still need to get vaccinated, right?
Absolutely. Absolutely. Yeah.
Yeah. You talked about herd immunity a little bit, so maybe we can just describe what that is and how that works.
Absolutely. Okay. Yes. Herd immunity. A lot of people have heard a lot about this as well. And I think the greatest thing to come out of this pandemic is just overall education and awareness on how diseases are spread. So, so that's one positive to take out of this. But yeah, so there's two ways for herd immunity to happen. One is through natural resistance, and the other is through vaccine resistance. So I think a lot of people are aware of the dangers, the obvious dangers in acquiring just a natural resistance. Obviously, death rates is a big one. We see with the Spanish influenza pandemic, where it affected over a third of the population with 50 million people dying. Obviously, that's not the result anybody wants here. So we're all really banking on hopefully getting a vaccine resistance. So when this herd immunity can happen depends on the virus that you have in question. And this comes down to the R naught value. So the R naught value suggests how many people each positive person can infect. So in the case of COVID-19, the R naught value is thought to be between 2 and 3. So if I had COVID-19, the idea is that I could infect anywhere from 2 to 3 people. Obviously, there's super spreader events and all of that, but in
Yeah.
general, that's what the R naught value is thought to be. So when we can acquire this herd immunity, we need at least 50 to 67% of the population to be resistant before that herd immunity can then kick in and the infection rates can go down. So that's the popular thought on that so far.
Right.
If you think of, I can't remember what the R naught value is for measles, but it's it's very, very infectious. So you would need a
Mm,
higher rate of immunity before that would be a herd immunity. And ultimately, the more people immune, the better. So there's people who can't get the vaccine, and it
right.
just becomes more and more important that as many people as possible can be vaccinated.
Well, and much like us wearing masks outside and where we go, right? Like that's protecting the people around us more so than it is protecting ourselves. There's a piece of that, but vaccination works a lot the same way and herd immunity works the same way, right? If there are people that can't get vaccinated, your community gets vaccinated, so you have less chance um, of getting that virus, right?
Absolutely. Yeah, it's very much I protect you, you protect me. And so
Hmm.
I'm wearing a mask to protect you. Yes, it provides me with some protection, but it provides more protection to the people around me in case that I'm infectious. It's the same with vaccines. I might not die from the virus, but I definitely want to make sure that I don't transmit it to somebody who can. So
Absolutely.
it's very important.
Talking, we can continue talking about vaccinations a little bit if you don't mind, because I know there are some concerns and misinformation out there about, vaccines aren't safe and they make you sick. And but specifically with the vaccines that have been developed for COVID are an RNA vaccine, correct? So they work a little differently. Is that right?
Yeah, so it's an RNA virus. It's a newer technique. I've talked about RNA being less stable than DNA, so that can be a little bit trickier to work with. So, um, especially with the initial vaccine that came out, a lot of people are talking about the difficulties with having to store it at minus 80 degrees Celsius.
Right.
So that can be a big reason on why a lot of people may have not used this technology in the past, but one of the benefits of working with RNA vaccines is that you can quickly make changes and quickly it was a reason why the RNA vaccines were the quickest ones to be developed and
Mm,
administered around the world is because you can engineer them so much faster. So the technology, is very similar with a lot of vaccines, but with the RNA ones, they were just able to be deployed a little bit quicker. And then
mm,
similarly, um, with a lot of the variants coming out, you can make changes to these vaccines a lot faster. So it's not a new technology. It's just it wasn't a technology that was used as frequently in the past. But you can see like with a company like Moderna, so they're using a very similar RNA technique, but their vaccine can be stored at minus 20. So they've
mhm.
been using that technology a lot longer than other companies. And so whatever, their secret is confidential. So I'm
Yeah.
not sure how they were able to stabilize it for minus 20. I know whenever I work with RNA, I've had to store them at minus 80 or else
Mhm.
it degrades to a point where it's not useful anymore. So whatever technology they've been able to come up with, I believe will be useful in the future so that we can make these changes. So using that type of technology then has not been limited to COVID-19 research. It's
Yeah.
started innovative research to other things. Like there's recent um, articles that have come out about the developments in malaria, a malaria vaccine that is using a similar technology. So past
Oh,
vaccines for
interesting.
malaria have not been as successful, but it's showing with the information that we've learned from the COVID-19 research that other viruses can be, or other diseases can have vaccines developed using a similar technology. So it's very exciting to see that emerge as such a useful technique.
What I find interesting about that is the RNA technology is so quicker, like you say, quick to respond and change. And I feel like that might aid us in future pandemics and responding to things quicker.
Pandemics have become increasing over the last 20 years. This is
Yeah.
the biggest one to come out, but I mean, we've had Ebola, H1N1, SARS. There's recent outbreaks of avian flu, H5N8 in Russia and India. So far, influenza has caused over 5 global pandemics. So this
Mhm.
has shown just how respiratory viruses have proven to be worth our attention, and the amount of effort that has gone into this specific pandemic, because really everybody was impacted by it,
Yeah.
will definitely help us going forward. This is not our last pandemic, but hopefully it's the last pandemic where we've had to respond in the way that we've had to. Hopefully we've learned enough,
Yeah.
we've developed enough technologies that we'll be better able to treat the next one that comes along.
Is there anything else you want to talk about?
I guess, one of the things that you'd brought up with was, advances in microbiology and
Yeah.
changes for the future. I mean, I guess I talked about um, specifically to pandemics and what we've learned, but the amount we've done in the microbiology field has been huge over the
Mhm.
pandemic. Just the amount of funding and resources and global efforts. I've been in webinars from people all over the world just saying, what technologies are we using? What's the most useful genomics tool for capturing COVID-19 genomes? There's resources that are open sourced online where the genomes
Mhm.
that I'm sequencing here for, the prairie provinces, I'm putting those genomes into a national or a global database so people can look and see, okay, what variants are circulating in which parts of the world. Epidemiologists are specialists that watch these movements of viruses, and how it's moving around the globe. And so they're able to use then these resources, from somebody even like myself, just in Regina, Saskatchewan, putting these resources in can be useful to people around the world. So that's really cool and really
That
exciting
is.
to me just to see those global efforts. And just the new advance. Like I said, I talked about the vaccine research is huge. Like I think this is
Yeah.
going to be a very useful tool for not just COVID-19 research, for a lot of things. I know before the pandemic started, not very many people were studying coronaviruses. So, for
Yeah.
myself, we have this other specialty in genomics, but then we kind of jump on the COVID-19 bandwagon and are a part of it. But after that goes away, we can use all of this funding and resources and knowledge we learned from the pandemic to our other efforts of research as well. And that goes for all the specialties
Mhm.
around the world. So that's huge. Um,
Yeah.
and then again, just the collaboration too that I hope will continue to be a thing when it comes to any future pandemics or global threats. So
Yeah.
we'll see how it goes, but it's exciting.
It is exciting. It's really exciting. Um, and it sounds like there's really groundbreaking research being done right here in Regina, which is also really exciting. And like you say, that it's being shared with on a global scale and like, yeah, that's very cool.
Yeah, I
Excellent.
mean, we're collaborating with people all over Canada, but we are doing efforts. We have collaborators from around the world as well that we're working with just here at the University of Regina. And then, like I said, we do a lot of collaborations too with like the Royal Saskatchewan Museum, and other universities. So not just the provincial labs, but we have active collaborations with universities in British Columbia, Alberta, Manitoba, Nova Scotia. I've even gone up to Nunavut to do some research with antibiotic resistance. So,
Yes.
yeah, the sky's the limit. I mean, we think we're just a small city, but we do make a big impact globally as well.
For the young scientists to be, what would you suggest if they're interested in microbiology to sort of pique their interest and sort of get down that path?
I mean, I'm a microbiologist, so I'm very biased, but we've done lots of outreach programs with Girl Guides and science camps to bring kids into the lab to just show them how exciting microbiology
Mhm.
is. So really, microbes are everywhere. Any
Yeah.
living thing interacts with microbes. Our body, I've heard somewhere that our body's 80% bacterial cells and 20% human cells. So if you think about
Crazy.
it, we're more bacteria than person. And those interactions um, ensure that we can be alive. We would not be alive without those interactions. So microbes get a really bad rap, especially, with the pandemic, that everyone thinks, ooh, bacteria and viruses, they're so bad. But in fact, bacteria especially plays such a huge role in human health. And without all of these interactions, we'd be very sick.
Yeah.
So I think it's cool. And just the amount of different interactions, whether you're playing, your kids are playing in the soil. You
Yeah.
can see interactions between the plants and the soil organisms and how those interact. Or, you see a bee buzzing by. We've done studies on that and what's causing the bees to decline. Are there pathogens
Mm.
or microbe interactions that are causing the health there? I mean, I've had one of my the PIs, one of the co-directors of the institute, used to bring his son in, and he went and swabbed my laptop, and there's all kinds of bacteria that grew and led to me, cleaning my laptop. But
Yeah.
really, a lot of that probably was not harmful, but it was cool that you could actually see these, under a microscope,
Mm.
or even just culturing bacteria, that there's this whole hidden world that you may know nothing about, but it plays such a significant role.
Yeah.
We had a debate the other day that all of the COVID-19 virus particles in the entire world could fit into maybe a Coke can, which led to us doing a bunch of math to whether it would actually be, a bathtub or a Coke can. But you think of like the idea that something so small makes such a huge impact to the entire world. And
Yeah.
this is just one organism. Well, the sky's the limit on which organism play a big role. And I think it's just fascinating. And so, um, yeah, I mean, whatever kids are interested in, there's microbes playing a role in whatever they're interested in. So whether they like cats or whether they like, you
Yeah.
know, trees or
Dinosaurs.
whatever it is, there's microbes that are involved in those interactions. So
Yeah.
yeah, dinosaurs, absolutely. So there's the sky's the limit for the questions that kids can have in that. And you can't be too young to be a scientist and be interested in that world. So that's my biased opinion.
That's awesome. That's great. I'm sure you're going to inspire lots of new people to explore the world around them. That's awesome. Well, I really appreciate taking your time today, Kara. This has been fascinating. I've learned a lot, so thank you so much. And
For sure. No worries.
keep doing the good work.
We'll try. Well, let me know if there's any other questions you have after, if you need help filling in any gaps or anything. I'm happy to do that. So,
I
but yeah,
appreciate that.
happy
That's
to
awesome.
be a part of it. I love the
Awesome.
science centers. Yeah, my kids love going there. Yeah, I'm happy to give back in any way I can, for sure.
That's awesome. I really appreciate it. Thank you so much. And yeah, go forth and have a great day.
Thanks, Lisa. Take care.
You too. Bye.